So we're right on time, and so I'd like to jump straight into the brief panel discussion, so 12 to 15 minutes. The way I'm going to structure this is I'm going to ask each of you a question in turn, and if there is time, I'll circle back, and then I'll give each of you a chance to say something very short as advice to the students listening in on the call. After the panel discussion, I'm going to bring in the questions from the audience. Okay. I'm going to go in the order of the talks themselves. So Bonnie, first question to you. I'd written this question before I heard your talk, and after I heard it, I really want to know the answer. How do you sustain a research program that relies so deeply on chemistry? Do you have a chemistry background yourself, or do you collaborate with chemists of different types?
So I have very little chemistry background. I was a biochemist, and then I became a geneticist, and I learned a lot of chemistry here. And I think, so I do have chemistry collaborators. I have a physics collaborator, chemistry collaborator, a structure collaborator, and a theory collaborator, and an engineering collaborator, right? And I think that's brain candy, right? And so then now the students and postdocs have all of those different backgrounds, right? Because there's more than me to advise. And so we're getting pretty good at it, at these different disciplines. But I guess what I would say is that, like, my goal is to train tomorrow's scientists, not scientists that were trained like me. And so they have to be open to doing all of these different, you want to go after the questions, not be limited by your training. And so I will say that, back to the chemistry part, is that these bacteria were talking with molecules, right? We knew that, and we could make mutants, and they didn't make molecules, but you can make mutants forever. It doesn't tell you what the molecules are. Chemistry does. So the science questions initially drove us, even though we were afraid, right, to do the chemistry. And then we started to succeed, and we're like, wow, now we can ask all kinds of questions. And so then we began to incorporate structure and physics and theory, right, for sort of the same reason. But chemistry was the first. And it was driven by the science questions that we couldn't answer based on our own training. And so I basically kind of knocked on the doors of colleagues. Princeton's a small school, so my chemistry colleagues are just across the street, so like a 1 minute walk away. And I kind of started knocking on the doors these were brand new molecules, so they captivated the chemist's attention. And it's been a pretty happy ride ever since.
So can I push you on that? I mean, you make it sound easy, but I worked in this interdisciplinary interface between my training in physics and biology, and I can tell you that the learning curve is steep in both directions, right? And one of the most difficult things is to sit and talk to a colleague, let's say in chemistry, and have that language where they're able to understand what interests you. And so, how long does it take before you're able to actually launch a real research collaboration? And how long just discussion over coffee?
Right. I agree with that. It takes real patience on both sides, and it takes hanging in there. Right. And so for sure. And so, now when we have lab meetings, it's out there, right? When we have lab meetings, right, for the new people, and it doesn't matter whether they're an undergraduate, a new graduate student, or a new postdoc, it's really hard for the new people because, when the geneticists get up and talk in all their 3-letter words, the physicists can't understand. When the chemists get up and they did this on a palladium, blah, 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 nobody can understand, right? But if we all hang in there, and then we're humble, and we say, and you have to be willing to say, I don't understand that. Explain that to me in simpler words, and then we do. And we're gentle to each other, and we're patient with each other, and then everybody comes along. And I think now one thing that's really, really good is that the new people are completely, they have imposter syndrome, and they're insecure because they can see that the physicists can make GFP fusions and the geneticists can code, but I think, like, sitting in that room, you can see those other people did it, and they were like me, right? They had whatever the training was, they were like me. And if you just hang in there, and if you are not so proud to say, I don't understand that helps. But you're right, it was slow going, and it didn't all happen at once, right? But then once, truly, and the chemistry was the start, once we started to do what I would call chemical biology, then we're kind of like, why not physics? Why not, we could answer these other questions. Why not engineering? We could answer these other questions. And so there was a sort of a, we got more brave over time at the, like, we can conquer this, if we have, and then of course, our collaborators have to be like that too. And then I'll finish. What I will say is that now, because my lab kind of has a history of that, students and graduate students, postdocs from all different disciplines come because they can see that there's a trajectory that will give them, there must be, they don't know what they're getting into, but there must be some kind of infrastructure in that lab, right, that allows it. And so then, it starts in the lab meetings, but then it all finishes over the benches, elbow to elbow, talking over the benches, drawing at, and teaching each other. And it's wonderful, right? It really, I mean, for me, I should have paid attention in organic chemistry, but I didn't. But now that I know it's because the bacteria talk, like I totally think I learned it. Yeah.
I love that answer. So humility and a kind of trust, and the people you're training are spreading this culture,
hopefully,
all over the world. Thank you. Zorana, the second question to you, and as I mentioned, and you also highlighted, this past 10 years has been a sort of golden age for these kinds of things. And it's a shame Ned Seaman is no longer with us, but he was really a pioneer in this area and inspired many of us to jump across these disciplines. So I want to ask you something about the subject I'm interested in, which is membranes, which don't feature in your approach to things. So if you add membranes on top of all these very cleverly functionalized particles, will you be able to enhance the set of things that are achievable in your theory and experiments?
Yeah, so that's an excellent question, because I think what you're trying to get is the point where we cannot achieve something that we're trying to do without the membrane. So that's the point that we are trying to get to, because we're doing all these very simple systems. They're sometimes actually even too simple that they can do something. But it turns out that, and we have some examples, that if you actually put a compartment or a membrane or something, all of a sudden things actually work. But we are not putting it immediately. We are trying to get to the point where we say, okay, there is no other option for this to work unless we actually have a compartment or a membrane or something that will condense us or channel us or something like that. So that's the idea. So it's really all from the bottom up.
So that's interesting. So you're saying that the set of challenges that you've sort of explored in your current systems are not complicated enough to require something like this 3D compartmentalization. So what do you think that new chemical challenge would be if it's not self-replication and it's not self-assembly?
No, well, okay. So I mean, okay. So the thing is, okay. So we can demonstrate some things, right? But it's all in numbers, right? So the numbers that we get, so how fast things are happening, how good they are, how efficient they are, they're really like child numbers compared to what actually can be we can see. And so I think this is really the point. It's not that they're not simple or more complex, and it's just that numbers don't match. And so numbers have to come from somewhere, and this is what we are trying to do. So even when we have a system where we can actually realize something, for example, we can make a catalyst, which actually is not that bad if you think about it, because there's absolutely nothing well, it's artificial in terms of it's artificial building blocks, but there is no any kind of driving. It really behaves like an enzyme, so it doesn't use energy. It's being recycled, so it sort of works. But then if you look at the numbers, they just do not work. You cannot get 10 to the 12 enhancement of anything. So the numbers come from somewhere, and many of these elements, like membranes and compartments, they have to be part of that.
But could it come simply from 3 billion years of optimization, and you've only been doing it for 10 years?
Sure, for sure. I mean, there is no doubt about that. I mean, for sure, this is a very important part. But yeah, so there are all these elements in the equation that contribute to the enhancement, and so what we're trying to do is disentangle. And again, the nice thing about these systems is that you can actually do kind of applying a kind of like evolutionary algorithms to try to optimize and to see how things are happening. And there are so many algorithms out there now that you can do all kinds of things. You can try to optimize for just some types of interactions, but you can also try to optimize for shape and such. So there are really, really like lots of things that that you can do. But yeah, we are doing it in steps, trying to see what, where the numbers come from. But we can only do it if we do it one at a time.
Great. Okay. Thank you. Gurmeet, I'm moving on to you. For first time, I want to ask a sort of science culture question. That's a theme that's actually been running through these life science across the globe talks. You alluded to, you mentioned Ayurveda a lot, and this is almost a sort of large monolithic systematic structure. Now, where does that intersect with the much larger body of folk knowledge in India or elsewhere? And how do you think these two different ways of approach, is it easier in some sense to bring Ayurveda into a fit with the kind of modern scientific techniques you mentioned, or is it that folk knowledge systems will also be amenable to that kind of approach?
Well, I think Ayurveda probably is a codification of a lot of the folk knowledge, right? It all started in folk knowledge. In fact, we have a database of medicinal plants and various used in medicine, probably the biggest such one, at least in India. And it has about 6,500 entries of different species, unique species, out of which 1,200 are 12 to 1,500 are Ayurveda. There are some from Siddha, some Unani, traditional Chinese medicine, some Swarikpa, which is the Tibetan system, but nearly 3,000 of them are not in any one of these codified systems, right? They are from folk knowledge, so references that we got from folk knowledge. So I think there is a great value, which is even beyond, which even Ayurveda has not tapped into yet. And that could be brought in. And so the modern tools, all of them have to be, I think what needs to be done is to approach these from a transdisciplinary framework with an open mind, and look at the molecular understanding and couple it with the systemic approaches to study these and put them together, right? Yeah.
And so following on that, if you imagine that these systems are actually embodied by oral traditions or within families and so on, and it's not really written down anywhere. One could imagine, and it's probably true, that we're losing a lot of that knowledge as we speak on a daily basis. In your opinion, is it likely then once you lose something like that, we'll be able to rediscover it using modern methods, or is there some sense in which the methods by which they were discovered are also lost, and so there's no way to go back and recapitulate those discoveries?
I think the methods by which they were discovered are already lost, probably. I mean, what we have are what the equivalent of what we consider as abstracts, in today's papers, right? The rest of the paper is gone. So we don't have that. So we are therefore, our job when we look at these is to try and deconstruct why is this formulation there, right? And how do we deconstruct? We deconstruct using these two approaches. I mean, largely the deconstruction had been so far from a simple reductionist point of view, but the last 10 years is all about the systemic tools which are coming up. The coming together of the multi-omics at low cost, the coming together of computational power, the coming together of new statistics tools. It's not just our traditional, but the Bayesian statistics and others are coming. So it's a coming together, and so all of these coming together is what I think is creating a new world, the possibility for a new way of exploring these molecules.
Again, it's a discipline which is ripe for investigation, and if you're a young person, this is the kind of thing that
I think one more thing, even the way we study things in clinicals, which are very strict inclusion criteria, to now free living studies, which are becoming possible, is going to be game changing. Yeah. Thank you,
Gurmeet. So actually, I'm going to leave aside the question for students because there will be other opportunities to do that later. Because I can see the stopwatch coming down. I'm going to go to the question and answer box and then come back later to other questions if there is time. So we have a few questions. What I'm going to do is I'm going to read them out to the appropriate panelists, and please do answer as you see fit. First is a brief one to Bonnie from Michael Weiss, who says, does one of the three quantum sensing molecules shown on a slide early in the presentation in fact contain boron?
It does indeed. Yeah. So that was a surprise, right? So that molecule, which is made as a 5-carbon precursor, the enzyme that makes it is called LuxS. All bacteria make the same thing, and it has cystiols. And the thing cystiols love to make adducts with the most is boron. And so remember, these are marine bacteria. Cholera, Vibrios are marine bacteria, and the ocean is loaded with boron. So the sort of instant that molecule gets made, it immediately makes this adduct with boron. And that double-ringed molecule that I showed, that is the autoinducer. So we have crystals of the receptor, and we can see the boron atom in there. And it was really fun to do that. Maybe this is I don't know who this person is, but if you're a chemist, boron has an incredible history in chemistry, right? And almost no history in biology. There's only like 3 molecules that are known to have boron, and they all have those cystiols. And so that was a real surprise to us. And we actually figured it out by making the crystal and seeing the molecule in it, right? Because we thought, because we got the mass of the molecule by mass spec, and carbon and boron only differ by 1. And so then we had to actually see it in the crystal, right? And that's how we got it. Yep, but it does.
Amazing. I'm going to read 2 questions together again for Bonnie before, since they're related. One from Lou Sheffer and one from Jeremy Cortez. The questions are, and I think you answered this in your talk, and the question may have been written before you answered it. So why do the bacteria that don't sense DPO make it? Seems like it's giving away info for free. And Jeremy asks, does DPO get metabolized or changed into any other molecule? Or is it assumed that DPO just stays in the environment without being changed?
Okay, I can answer them both together. I don't know. And so the first answer, so with slightly more color than that, for the first question, I don't think the bacteria making it and giving it away, they're not making it because cholera is going to be invading. I think that those microbiome bacteria, other bacteria that make DPO, presumably some or all of them use it, and we just don't know what the receptor is, right? It's not VQMA, right? That I showed you. So what we're trying to do is to squirt, we can synthesize DPO, we got buckets of that stuff. We're just squirting them on cells and trying to do RNA-seq and see what is the regulon that's controlled, and hopefully we'll find the receptors. That's a task for us. The answer is I don't know, but I don't think they're giving it away. I think it's a signal molecule for them as well. Fantasy until shown differently. And then to the second question, we're trying to figure that out now. Does DPO get modified more? Does it fall back apart? It doesn't fall apart because enzymes put it together, but it's a very stable molecule, at least under lab conditions. So we guess it hangs around, right? But unproven in realistic, it certainly hangs around in a test tube, with a bunch of bacteria in it, unproven, in an intestine or any real biological system like that. But that's certainly on the list of things we're trying to figure out.
Thank you. Next question to Zorana. Ashish Bihani asks, what do you think about the role of intrinsic disorder maintained by living systems in order to form complex assemblies? Would simulations based on energy minimization be able to incorporate these possibilities?
So, I mean, we haven't done anything on that. So I think my answer is going to be underwhelming. I think any kind of exploration and using simulations to do this, I think it's welcome. They can, even though we do a lot of approximation in simulations, they can still give us an insight on how to try to understand how things work. So I'm sorry, Ashish, but I cannot say more than that. I haven't done anything.
Could I extend the question a little bit? So, he mentioned energy minimization. So in a sense, if you think of equilibrium as having a free energy minimization principle, how many of your systems are actually out of equilibrium, and that's essential in order to make them do what they're supposed to do?
Yeah, so most of the self-assembly stuff is equilibrium, so it's not out of equilibrium. For the replication and catalysis, those things definitely use up energy. So for that one, you need energy inputs. For purely catalysis, that doesn't require energy input. Folding, there, at least because this is very, very artificial system, because we are completely manipulating which pathways are taken in the folding. This is very, very much out of equilibrium. So yeah, so it really depends on what we are trying to do. It's not always one or yeah, it's not clear which one is best. I mean, for example, with the folding, in the folding case, we do both, because we want to understand also the just the equilibrium distributions and how we can manipulate that we don't get them and how we can destroy them, right? So we do both, but it really depends on the system.
Okay, thank you. A question for Gurmeet from an anonymous attendee. When you think about plant-based therapeutics, don't you think that not a single molecule is responsible, but multiple active molecules are in there and responsible for their action?
Yeah, I mean, definitely. And that's the systemic angle, and not just one plant and multiple molecules, but also that formulations have 5 plants and multiple molecules from all of them. So why are they all together? So we do need to study them together. So the approaches of phytoprofiling and then the metabolomics together, studying them together and trying to understand this whole area, and then the microbiome mediation in between. I think those are some of the approaches which will help. But yeah, definitely, it's at least the approach from Ayurveda points to multiple molecules together and not one molecule, one benefit, right? One target. Yeah.
And staying with Gurmeet, Ashish asks, says Al-Biruni once commented on Indian knowledge that it is a mix of diamonds and stones. This is an ancient Islamic scholar, and critical analysis is needed to sort it out. However, this analysis did not come about, and a lot of rational systems died out. How can we maintain rational schools of thought without purging plurality of opinions?
Yeah, no, I think that's true of all knowledge systems, that there is a lot of noise, and I think therefore we scientists are in job to be able to decipher the noise from the rest. Otherwise, we could all have been on a WhatsApp university. But yeah, I think it's a challenge. And if you go back to, I mean, most knowledge systems have been fairly open. Maybe we've over time kind of closed them and gone towards maybe more faith opposed to more questioning and creating insights and hypotheses and moving forward with it. But I think like all knowledge systems, a lot of the Indian knowledge systems, their foundations have been very open. I mean, you look at the ingredients in there and you can see from one text to the other, if you look at the chronology, more and more ingredients have come in, formulations have changed. The example that I gave you about stabilization of butter oil comes from not from the original text, which had a very different formulation, but from a 15th, 16th century text, which is very recent, compared to when the Ayurveda was founded. So there have been improvements and things have been weeded out. So it's not that it's not being done, but needs to be done way more probably. Yeah.
Thank you. And there's also a question about whether a reductionist approach will actually work, and I'll just leave that in the chat box, and maybe you can answer it just in the interest of time, because I want to give all 3 of the panelists now a chance to give their message to students. So Bonnie first, then Zorana, then Gurmeet.
Okay, so let's see, I'll say one little tidbit, which is I think sometimes young scientists or people thinking to go into science, they confuse challenging with I'm no good at it. Right. And so, you get a B on your test, or your experiment doesn't work, or you find the puzzles really hard, and then I think that sometimes we scientists think maybe I don't have any ability here. And I would say that difficult or challenging and lack of ability are totally different things, right? So first of all, you want it to be challenging, right? Because otherwise you don't have anything to do. And also the easy mysteries already got solved. So you want to be studying the fundamental big mysteries. And so I just hope that the students who are listening, don't get beaten down or drummed out of it. It's the greatest life in the world to make discoveries, to be the first person to ever have a thought about something ever in history. Meaning that's an amazing life. Right. And of course, it's challenging and difficult, but that's the fun of it. And so don't confuse that with maybe I'm no good at this.
Thank you. Zorana.
Yeah, so that's a beautiful message, Bonnie. So what I will just add on that, because I think that's it's partly what I was planning to say. I would just add that you should always pursue to talk and work with people that you like that you enjoy talking to, because they don't have to be big shots and big universities. It's really not about that, because the synergy is really the thing. And if you really like someone and enjoy talking to someone, ideas and things will just come out because you're in sync, and then that's the whole point, right? So it's they don't have to, I mean, just you're just more together than you are alone, and it's important that you like each other and you enjoy doing that. So I will just add that to what Bonnie said.
Yeah, so not to forget, science is a human activity, and I think the last 2 years have been a real aberration, and we're all itching to get back to the way we used to work.
Sure.
And Gurmeet, you have the final word.
I think, building on, I think the key is to be open, and listen to many different perspectives, listen to many different voices. Yeah, I think in this openness, life is certainly about reductionism and choices. No, evolution, if you look, is all about choices. We know small differences can create so much of havoc. One amino acid change can create sickle cell anemia from nothing. Once amino acid change created the Delta variant, right? It was all, it's a very small change. So life is about becoming specific, but this reductionism, this specificity happens in a network, not alone, right? And therefore we need to understand the whole network as well, the various pulls and pushes. And it's all about talk. And I loved, just building on from Bonnie's talk, right, where it's all about communication and how molecules communicate. No, we thought of food initially as fuel, right, calories, and then we thought of it as, we think about it as nutrition, right, micronutrients, vitamins and minerals. But today we also talk about food is information, right? It is the molecules in there are communicating with the DNA through our epigenome, right? So it is deciding, it's like the execute button on our computer and deciding which part of our DNA will execute, right, and express. So this openness to various ways of thinking and communicating, I think those themes run parallel, not just in our test tubes and our model systems, but with the community as well, right? I think that's when food becomes joy. Yeah.
Thank you, Gurmeet. So I want to end by thanking all the speakers. You put these things together and you hope it'll fly. And I think today really flew, because there's a common theme that emerged between these three talks that sounded very different from their titles, but really that chemistry theme that holds everything together really shone through the talks today. I also want to thank Janelia, Ron Vale, thank Janine for the opportunity to host these talks today. And this is such an amazing platform. The talks are recorded. People can see them all over the world, either today live or later. And I know a lot of people make use of that opportunity. So before I hand back to Janine, I just want to say that for the trainees who are on in the audience, Ulrika is going to lead a session immediately after this, where you'll get to meet and chat to the panelists, and she'll explain how that works. So thank you, everybody, for an amazing session.